Good afternoon, everybody. My name is Lisa Lin. I'm one of the speakers for today's webinar. I'm in the Office of the Director, Center for Biologics Evaluation and Research within FDA. Nabil Al-Humadi, one of senior reviewers of CBER, and Susan DeHaven, co-leader of Send for CBER team, will also present in this webinar. At today's webinar, we will introduce to you what you need to know about Send for CBER. SAND stands for Standard for Exchange of Nonclinical Data. We will introduce CBER's requirement and the support on SAND. We will introduce some of the important study types and data points for CBER nonclinical studies. Get to know the perspective from reviewers' point of view and further understand the future development on SAND for CBER. Does anybody know what's CBER's requirement date for SAND? I hope you are all aware of recent announcements on CBER's requirement on SAND. In July 2020, CBER has added the CDISC SAND Implementation Guide version 3.1 on FDA Data Standards Catalog for nonclinical data standards, with the requirement date for March 15, 2023. As you know, FDA Data Standards Catalog listed all the data standards and the terminologies that FDA supports for use in regulatory submission. In addition, the FDA has the statutory and the regulatory authority to require these standards listed in FDA Data Standards Catalog, which means FDA will not accept an electronic submission that does not have study data in compliance with the required standards specified in FDA's Data Standards Catalog. Evaluation and testing of the SAND standard for CBER is one of the important projects listed in FDA Data Standards Program Action Plan. Through a couple years of strong and well-resourced collaboration between CBER review team, CDISC SAND team, and industry volunteers, the team has assessed special needs from CBER nonclinical reviewers, performed the gap analysis between reviewers' needs with current SAND implementation guide. The team has also performed the proof of concept pilot on multiple studies submitted from industry, and made robust recommendations for CDISC on future standards development. And now, we are ready to implement SAND for CBER. CBER has also published the Federal Register Notice in July 2020, announcing the support and the requirement for SAND. Here's a screenshot of the Federal Register Notice, and on the left, you can find the link of the Federal Register Notice website. Please feel free to click on the link to read when you have a leisure moment. CBER has 3 review offices reviewing all CBER submissions. They are Office of Vaccine Research and Review, OVRR, Office of Tissue and Advanced Therapies, OTAT, and the Office of Blood Research and Review, OBRR. This gives you some overview on what products OVRR reviews. OVRR mainly reviews vaccines for prevention or treatment of infection disease indication, allergenics products, and etc. OVRR has a lot of nonclinical study and data types which are fit for SAND model, like single and repeat dose toxicology, developmental and reproductive toxicology, genotoxicity safety pharmacology, immunogenicity, and biodistribution studies. These study types are all well aligned with SAND implementation. Some of the important data points the team has identified are timing of endpoints following vaccinations, like 24, 48 hours, and 7 days post-vaccination. DRAIS scoring, body temperature, acute phase reactants, immunogenicity, serology assays, injection site histology. All these data points are very important to reviewers, and we hope these important data points are all included in future SAND data submission. For OTAT, they review a lot of cutting-edge products, including stem cell products, gene therapy products, and etc. There are a couple non-clinical study types are well aligned with SAND implementation, like proof of concept safety, toxicity, and immunogenicity therapeutical vaccine studies. Immunogenicity data is very important to reviewers. SAND for CBER team has been working closely with CDISC on developing IS domains for non-clinical immunogenicity data. OBRR is an active participant in the project group, but SAND doesn't have any near-term impact on OBRR non-clinical studies. The SAND implementation on OBRR non-clinical studies will be case by case, especially for GLP-compliant toxicology studies and carcinogenicity studies. We recommend sponsors to contact Review Division to discuss the specific submission data requirement. At this point, CBER reviewers are ready and looking forward to SAND submissions. Currently, CBER reviewers have been provided CDISC study data standards training to have basic knowledge and familiar with the standardized study data. They have also been provided training on the analysis tools, including JUMP, JUMP Clinical, and SAND Explorer. Some of the tools provide a visualization of SAND data for them to look for patterns and trends in preclinical data. CBER eData team provides consultation support service to CBER non-clinical reviewers on how to review SAND data by using the existing tools, providing data standards validation check to ensure the quality of the submission data. CBER reviewers has conducted 4 valid proof of concept studies so far. Reviewers had opportunities going through the practice of data standards validation check, looking at defined.xml file with data, and using non-clinical study data reviewer guide to help them with data review. They also had hands-on experience by using tools to do data review and analysis. Next, you will hear from one of CBER's senior non-clinical reviewers, Nabil Al-Humadi, giving you his perspective on how SAND data impacts his review on non-clinical submission data. Thank you. Nabil, please.
My name is Nabil Al-Humadi. I am a reviewer at the Center of Biologics at the FDA. Today I would like to talk about the SAND importance in the view of a reviewer like myself. And I'm going to talk about both time and precision and how they are important to us. This is a disclaimer, this just to let you know that these are my views and not the FDA. What is most important for reviewers to complete their job? This is a very critical question. To review a large amount of data requires time. This time is required for accuracy in summarizing the findings to reach the right conclusion. This is very important. Time is very critical when it comes to an emergency situation and non-emergency situations sometimes. Emergency situations, the best example is what is going on with coronavirus pandemic. Instead of 4 weeks to review, I was given only 4 days. In non-emergency situations, when we have multiple applications in our hand, like 3 new INDs and 1 BLA. Sometimes the BLA includes more than 10 studies. At one time, we received 16 studies in 1 BLA, even though they were distributed between 2 reviewers. We ended up with 8 for each one of us. Still, this is a lot of studies to review. And in this kind of situation, submission of the data in an organized, easy to evaluate way is very important. One of the most important features of SEND is that the reviewer will receive the data in very organized, easy to review modules. What are the types and amount of data we receive to review? In my next few slides, I'm going to show you to answer this question. Study designs, just to give you an idea about the size of the studies, might include 2, 3, 4, or sometimes 8 groups. And this is for both males and females. And we have multiple sacrifices, sometimes more than 2 sacrifices. Most of the studies includes main study sacrifice and recovery sacrifice. Again, it depends on the type of the study. It might go more than 3 or 4 or 5 sacrifice time points. This is a huge amount of data. I apologize for this slides because it is busy and I'm not expecting you to read all of it, but I'm going to go over it very quickly to show you what we receive, what kind of data we receive. We receive data related to mortality, post-dose observations, cage site observations, detailed clinical observations, dermal scoring, body weights, food consumption, body temperature, ophthalmic examinations, and we receive data related to clinical pathology evaluation, which includes hematology, coagulation, clinical chemistry, and one of the important biomarkers, C-reactive protein. And from this slide, you can see there are 3 time points for terminations. Day 3, day 7, and day 15. Hematology parameters includes a lot of data related to red blood cell, white blood cells, clotting potential, and these the parameters we look at it is listed on the right side of the table, and sometimes it includes bone marrow cytology too. Serum chemistry parameters include carbohydrate metabolism, liver function, which includes ALT, AST, ALP, and bilirubin, acute phase reactants, which these are biomarkers for inflammation, includes fibrinogen and CRP, kidney function, others related to albumin, total protein, carbon dioxide, globulin, AG ratio, etc. These are all listed on the right side. Immune responses. We have to look at all the data related to anti-drug antibodies, which includes IgG, IgA, sometimes IgE, and at 2 different time points here, like day 3 and day 15 sacrifices. This is to evaluate the difference between the treated groups and the control groups, and to see if there is any difference between males and females in their responses. Organ weights. We see there are a large number of organs will be collected at sacrifice, and we have to look at the differences between the treated groups and control groups. A huge number of tissues will be collected for histopathology examination. This has to be looked at for each group and for both males and females. And for the previous slides showed you the general tox studies. In the next few slides, we're going to show you the developmental and reproductive toxicology endpoints. These are all related to mating period, like mating and fertility indices, gestational period, includes maternal evaluation, fetal and placental weight, gestational period, which includes fetal external evaluation, fetal physical evaluation, fetal skeletal evaluation plus ossification sites, and also includes the lactational period, like litter evaluations, lactation and survival indices, pub developmental landmarks, immunological endpoints in DART studies will be looked at in the serum antibody determination for F0, F1 fetus, F1 pup generations. Additional assessments, which include, this is case-by-case basis, if vaccine-induced adverse effect, like histochemical analysis for antibody depositions, neurological assessments, this is for safety pharmacology studies, immunological endpoints in developmental immunotoxicity study. How could SEND help us? SEND could help us in organize the data, help us review the data in an efficient way, and provide a tool to create tables and graphs to present the most important findings, especially when it is linked to JUMP or SEND Explorer. In the next few slides, I'm going to show you some graphs which was done by SEND Explorer. This is body temperature, and this is only for two groups for males and two groups for females. You can imagine if I have four for males and 4 females, or 8 groups for males and 8 groups for females. But in this case, I can see that if there are any temperature above 40 degrees, which it gives me immediately the full picture about the body temperature and if there are any adverse effects related to the test article treatment. In this graph, body weights, we can tell the difference between both treated groups and control groups in males and females. And this is for creatine kinase levels. Again, it is very important to see from one look, we can tell the difference between the treated group and the control. C-reactive protein is one important biomarker for inflammation. And from this slide, it is very easy to see if there is any level of C-reactive protein above 40 mg per liter. To tell me if there are any kind of inflammation in any group. Neutrophil levels, the difference between both treated and control groups. We can also get tables for any microscopic findings. This is very important. It can tell me very easy the difference between the treated groups and the control groups in their findings, and how many animals and the severity of these findings. Finally, this is a graph about the microscopic findings in different organs like kidney, liver, spleen, lymph nodes, and site of injection. It is very easy to find out what is the difference between the treated groups and the control groups and the severity of the finding. Bottom line from my presentation, that SEND will help me save time during my review and present my conclusion in a precise way. Thank you.
Hi, I'm Susan DeHaven from Sanofi, and I'm also a co-lead of the Send for CBER team along with Lisa Lynn. And I'm going to talk to you a little bit about the Send for CBER team, as well as the proof of concept pilot that we conducted earlier this year. The Send for CBER team key activities included a deep review of the SEND 3.1 IG, followed by the proof of concept pilot that we'll talk about today. And through the end of this month, we'll be publishing our assessment summary report on the CDISC Wiki. I want to let you know that all of the donated study data, along with the team analysis notes, are posted on the CDISC Wiki, and there'll be a link to that at the end of this presentation. Following our pilot, FDA did move forward with submitting the Federal Register notice that Lisa described to you earlier in this presentation. The SEND4CBER team is a strong collaboration between FDA and CDISC SEND experts, and you can see they're coming from organizations across the spectrum of stakeholders who are interested in submission of electronic data to the FDA. We received 4 proof of concept pilot studies that were in 2 modalities, 3 vaccine studies and 1 gene therapy study. 2 of the studies were in monkeys and 2 were in rabbits. We did not receive any study that would have been suitable for an OBRR submission. However, the reviewers did participate in the review of all the domains of these 4 studies along with us from OBRR. This is a list of the endpoints that we received on these 4 studies. So not every endpoint possible was submitted within these 4 studies, but we did get a nice cohort across the numeric as well as text data types. And I made this grid in order to map for you the individual endpoint to the domain that it was submitted in for each study. And you can tell here that not every endpoint is received on every study, but overall, that of course is normal, depending on the protocol and the methods section and which endpoints are measured per study. So this gives you a little picture of the SEND package that we received for each study. Each study did include study design domains, which I did not list here, but they are also posted on the wiki with the data set, as well as the non-clinical study data reviewers guide, the defined file, and the study report. I want to specifically point out to you the use of the IS domain in 3 of these studies. IS is for the immunogenicity specimens domain, and it was included in the pilot as a custom domain based on the clinical SDTM model for immunogenicity specimens. It is not yet in the SEND standard, and there's currently work going on within CDISC in order to evolve the IS domain for non-clinical use. And you can see here 2 of the studies used IS to report antigen level endpoints, and 1 study used it to report ADA endpoints. So the pilot outcomes, we had some very specific study findings considerations. Overall, the reviewers were able to get a quick overview of the data and were easily able to see differences between scheduled measurements across time, able to identify outliers and determine key time points of collected data relative to the dosing. They did mention that correct mapping to controlled terminology and consistent units within the SEND dataset, as well as between the SEND dataset, the study report, and the NSDRG is very helpful. Use of the variables in the SEND data needs to be consistent with the SEND IG definitions for those variables. Reviewers noticed that in order for the utility of the data to be very clear, there needed to be a very consistent relationship between the use of the data in the variables and the definition of that variable within the SEND IG. Original result values, what we call the ORES values, were very useful to the reviewers. They like to see the full text of the observation as collected. You know, if you've used SEND data before, that ORES can be parsed into its parent domain variables, and there might even be comments associated with that, or also supplemental qualifiers that can be sometimes difficult to reconstruct if the full observation isn't also documented in the original result. And reviewers were able to use supplemental qualifiers in order to make the relationship between assay result data that is reported in summary in the study report and the individual results that reported below limits of quantification. Supplemental qualifiers provide the quantitative values that were used in the study report for those below limit of quantification values. That reviewers found quite useful. Some overall package considerations, we would call the overall SEND package that came out of the pilot. The study designs for single and repeat dose tox studies for CBER fit into the SEND trial design domains quite well, although the reviewers found that study design descriptions in the NSDRG are very helpful. Reviewers found important information dependencies within the NSDRG and also across the define file. So the SEND package includes not only the data sets, but the NSDRG and the define, and they appreciated clear description of differences between the SEND data set and the study report to be included in the NSDRG, that the information in the define file is consistent with the data set content, and that any extended terminology that is used in the data set is explained in the NSDRG. And I would say that if anybody has experience submitting SEND to CDER at FDA, you'll probably realize that these are very consistent observations between the two centers of FDA. And some other considerations that we'd like to point out. The concept of what I would call a hybrid situation, where electronic data submissions may also have data endpoints within that submission that were included in the study but are not providing electronic data. This is possible. Non-clinical studies submitted to CBER can include some endpoints modeled in SEND and other endpoints that were in the study but are not modeled in SEND. And this is really the result of what Lisa described earlier, in that not all endpoints for CBER studies are yet in the scope of SEND. And even though that's true, all the reviewers unanimously agreed that all data that can be submitted in SEND is helpful for the review. So certain data like biodistribution data that can be modeled using a PC domain, even though it's not specifically mentioned in the SEND IG, including it in the submission is very helpful for the review. Also, we talked about the IS domain. This custom domain is not required, but it can be accepted. Or data such as ADA might also fit not just in a custom domain, but it might fit in the LB domain, which is a standard SEND IG 3.1 domain. And in fact, the FUSE non-clinical working group has published a white paper on exactly how to do that. Overall, it's important to clarify for the reviewers for each study which data has been provided in an electronic format and which not, and the NSDRG is the place to report that. So we know as future SEND IG versions come into publication and adoption by CDER, the scope of electronic data that can be included in a SEND data set is expected to expand accordingly. So the SEND for CBER team future, we consider ourselves involved in an ongoing mission to support the CDIS SEND team to include CBER considerations in their standards development, and also to support FDA data standards efforts. We do this by participating in domain working groups. Right now, we're very active in exposure, immunogenicity specimens, and modeling of dermal and ocular findings. We contribute to relative controlled terminology development, and also to decisions on what will be included in the next version of SEND, which we expect to be called version 3.2 at the moment. And we will remain engaged as future SEND IG versions come into consideration, development, publication, and adoption by CBER, so that we can continue this expansion of scope for the types of data that they can receive. For example, repro studies in the future could be submitted to CBER, and this will be consistent with the DART IG that is currently under consideration by CDER, but is not yet on the Study Data Standards Catalog. The team meets about monthly now to support FDA data standards efforts by developing recommendations for a technical conformance guide to be consistent with CDER, or if there's anything specific for CBER that might be submitted for the guide, we can develop those recommendations. We also consider the conformance and business rules that will be applied to the CBER e-data submissions, and we'll continue to communicate the proof of concept pilot outcomes and other team deliverables to industry stakeholders. So resources available for you are here listed, the links to these places. FDA Data Standards Catalog is the place to find which standards are currently supported by FDA. The link to the Federal Register notice that Lisa described earlier. There's also a link to the FDA Data Standards Program Action Plan, which outlines the strategy of FDA for accepting data standards, and our Send for CBER wiki site link, which is where you'll find all of the pilot data and the deliverables from the Send for CBER team. So in summary, CBER is ready now to support send data for non-clinical study submissions. The requirement date for CBER send submissions is March 15th, 2023, as set by the Federal Register notice. And the future development on the Send IG for CBER considerations will continue through the work of the Send for CBER team in collaboration with CDISC. And I believe we'll be taking questions at the end of our presentation, but if you'd like to submit questions in writing to FDA, you can use this email address here, cber-edata@fda.hhs.gov. Thank you very much.
Thank you all for those great presentations. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We have a first question coming in. This one is directed to Lisa. When are you required to submit send data?
So as we stated in the federal in FDA Data Standards Catalog, the requirement for send the data is March 15, 2023. That's for the study starting date, and the applicable study types as described in Send IG version 3.0 and Send IG version 3.1. And those are listed as in the Send IG for single dose toxicity, repeat dose toxicity, and carcinogenicity data, and also for the cardiovascular and respiratory for the safety pharmacology studies.
Thank you for responding to that question. We have another question for Lisa. Should all animal preclinical regulatory studies be submitted with SEND format, including safety farm study?
Yes, so as I answered the first question, that covered this question as well. So basically, you know, Send IG version 3.0 and 3.1 are supported by CBER. So all the study types apply to Send IG version 3.0 and 3.1 are the requirement as the study type.
Thank you for responding to that question. We have another question for Lisa. Does CBER expect SEND datasets for legacy non-clinical studies if a marketing application is filed after March 2023?
So the requirement applies to the study start date. So if as long as the study started before March 15, 2023, you don't have to submit SEND format, but we still strongly recommend if you can have SEND data to submit to agency, because as Nabil presented in his presentation, it really helps the reviewers to review the application with data.
Thank you for responding to that question. The next question is for Nabil. Are there any differences between experienced and unexperienced pharma companies in the regular IND submission?
Of course. Yeah, we have seen huge differences between experienced and non-experienced pharma. We try to help as much as possible in the pre-IND meetings, which unfortunately, some non-experienced pharma, they don't contact us for this valuable meeting. But yes, there is a huge difference, and data coming through SEND will eliminate this difference. Thank you.
Thank you for responding to that question. The next question is for Nabil. Will SEND submissions eliminate these differences?
As I mentioned in my response for the first question, yes, that will eliminate, because every single pharma company will use SEND modules, and these are very clearly stated. And when you use any kind of serology data in the IS domain, it will be easy for me to find this kind of information, and I know where it is going to be located. So when it is standardized, it will definitely eliminate these differences.
Thank you for responding to that question. It seems like we had a little bit of technical static, so we're going to move on to our next presenter. The next question is directed to Susan. Were there any major differences between CBER and CDER needs regarding SEND?
No, there haven't been any major things identified. Several times during our assessment, questions would come up, and we would refer back with CDER to understand if we really had a difference between CDER and CBER interpretations of SEND or uses of SEND or not. And each time we did that, we found that the reviewers were actually quite aligned. And an example of that is in the exposure domain, where we are now participating in the next version of the exposure domain development. So the answer is really no, not so far.
Thank you for responding to that question. The next question is also for Susan. Were non-animal-based endpoints considered in the SEND for CBER assessment?
Not so far. During the review that we've done in this past effort, we did not consider any non-animal endpoints yet. CDIS is developing some in vitro endpoints through GeneTox right now,
but we are not assessing that, and they're not included in the requirement yet.
Thank you for responding to that question. The next question is for Susan. Can someone still join the Send for CBER team? If so, how?
Yes, we'd love to have new members on the Send for CBER team. You could reach out to either Lisa or I, send us an email, and we'd be glad to onboard you to that team. You could also reach the Send for CBER team through the CDEX Send team. So there, all of our email addresses are variously throughout the CDEX wiki, and hopefully you'll be able to find us.
Thank you for responding to that question. The next question is addressed to Lisa. Will the technical rejection criteria be updated to reflect the implementation dates for CBER?
Yes, definitely, because the technical rejection criteria applies to both CDER and CBER. But since the requirement date for CBER is not until March 15, 2023, so it won't be updated at this time, but it will definitely be updated for future.
Thank you for responding to that question. The next question is directed to Lisa. Is, for example, Send Explorer available for sponsors?
Send Explorer is one of the tools available for FDA reviewers. I believe that's a marketing, I guess, tool available on the market. So I'm sure you can contact the vendor for purchasing Send Explorer, but this is just used as one of the tools for CBER and the CDER reviewers on the non-clinical stuff.
Thank you for responding to that question. We have another question for Lisa. If I have a biologic under review and a division within CDER, does the CDER Send implementation timelines apply, or do CBER timelines apply?
So the requirement goes to each center. So if it's a CDER BLA, it goes with CDER timeline. So definitely refer to the CDER review division and according to CDER timeline on the Send requirement.
Thank you for responding to that question. The next question is directed to Susan. Where could I find the list of studies that need to be submitted in Send format?
Okay, so this is a great question. The Send IG 3.1 outlines the study types. So as Lisa mentioned earlier in her answer to one of her questions, of the single dose, repeat dose, carcinogenicity, respiratory and cardiovascular safety pharmacology studies at a high level. The next level is, as we know for CBER studies, because of the there's a variety of what we would consider traditional toxicology endpoints, as well as other endpoints that are, let's say, maybe not usually in a CDER type study. In those cases, it will be important to discuss with the reviewers at the pre-IND meeting which data you're able to put in Send format and which not, because we do expect that you could have a study type like a repeat dose tox study that is in scope, but data within that study that is not in the scope of the Send IG modeling capability. So the study type question is I can really only answer at the quite high level, but certainly I believe at the pre-IND meetings or in discussions with your reviewers, you can really get a more clear answer study by study.
Thank you for responding to that question. The next question is also for Susan. Could you please confirm that SEND is not required for DART on preventative vaccine?
Okay, so the DART implementation guide is published by CDESC, but it is not yet adopted on the Study Data Standard Catalog. In order to submit DART studies, you would need to use the DART implementation guide models, and since they are not yet supported by FDA, they are not required.
Thank you for responding to that question. We have another question for Susan. The endpoints measured are traditional toxicology endpoints, many of which are not affected in cellular gene therapy products studied. Can we assume that other endpoints, such as VCN, chimerism, etc., are not going to be required to be submitted in SEND format?
Yes, you can make that assumption. I think as we evolve in the Send for CBER team, I can tell by the questions here, this is an area where I think we would like to explore some more clarity to be able to be published to the industry. But yes, there's definitely endpoints we understand and reviewers understand that will not fit in SEND, so cannot be required to be submitted and sent.
Thank you for responding to that question. The next question is for Lisa. Regarding sample SEND data submissions, if a sponsor has successfully submitted SEND data to CDER, is a CBER sample submission required or advised?
No, CBER doesn't request any sample data. That's different from CDER. You know, we only accept the real submission data.
Thank you for responding to that question. The next question is directed to Lisa. I understand that if applicable non-clinical data are to be submitted, it will have to comply with the SEND data format after the effective date. My question is that whether data are required to be included whenever the non-clinical study report is submitted for any purpose during the IND stage.
Yeah, so usually, yeah, we do request data submitted with the study report. If there's any challenge or any difficulty, but you can discuss with review division.
Thank you for responding to that question. The next question is directed to Lisa. If you submit a legacy data set, can you use simplified trial summary data sets?
Yes, so once the requirement starts, I guess, be added to the technical rejection criteria, you should follow exactly the same requirement on the technical rejection criteria, which includes the simplified TS XBT.
Thank you for responding to that question. The next question is directed to Nabil. What is the difference and time between finding the information in regular submissions and in SIN submissions?
There is a huge difference. As with SEND data, we can locate our data very easy as we know which module is belong to what data. And this is very important. When we are on the same pages, industry and the FDA on the same page, then we will be able to find out the data in very easy time. And to tell you the truth, it might be goes down to 1/10 of the time needed in regular submission.
Thank you for responding to that question. The next question is for Nabil. Can we create graphs or tables when data is submitted regularly, and what is the difference in the ways to create graphs or tables between regular and SIN submissions?
Regular submissions is almost impossible to do graphs and tables for us because we have to transfer the data from their original submission to our softwares, which we need, whatever software is needed to create the graphs. In SEND data, all what it needs, few clicks. And this is very helpful for us. I can create graphs and tables with SEND submissions within 10 minutes. And I did that in some of the early submissions. And this is the huge difference between regular submission and SEND data submission. Thank you.
Thank you for responding to that question. The next question is directed to Nabil. Will SIN give an advantage to unexperienced and small pharmas in submitting INDs or BLAs?
Of course, that's for sure, because they are going to know exactly what they are going to do when they put their data in their own modules. This data goes to vital signs. This data goes to serology data in IS domain, and they will definitely, like SEND data, it's going to direct them where to put their data in. And this is, it's going to give them more experience in a very timely manner. And but all what they need is just to participate in our meetings, and they need to know all about the modules we are dealing with. It will be great if they can contact Lisa or Susan to get involved in this kind of meetings. It's very important. Thank you.
Thank you for responding to that question. The next question is directed to Lisa. Who should we contact for submission questions?
Yes, as we presented here, please send any email regarding to CBER submission to cber-edata@fda.hhs.gov.
Thank you for responding to that question. The next question is also directed to Lisa. What are the documents we need to pay attention to on the FDA website regarding data submission?
So on the FDA Data Standards Resource website, there are a couple important documents as we listed in the resource page on the presentation. One is the FDA Data Standards Catalog. That's updated as it goes. If any standards accepted supported by FDA, it will be listed on the FDA Data Standards Catalog. Then there's also FDA Study Data Technical Conformance Guide, which is updated twice a year in March and October. Any special requirements, recommendation from reviewers regarding to study data submission will be updated.
Thank you for responding to that question. It looks like we have time for just one more question. And this question is directed to Susan. Where can I find FDA comments on the pilot study?
All of the FDA comments, the notes that we took during our review meetings of the pilot studies are posted on the CDISC wiki. They are a bit real-time looking. They were not massaged or changed or edited really. It really captures the real-time review that we performed during the pilot, and I think they're interesting. And if you have questions, as Nabil said, join us. We're happy to explain more.
Thank you for responding to that question. And also, thank you to our presenters for your very informative and timely talk, and also for responding to the questions that came in. We have just a few closing reminders.